Start recording. There's our join code. I presume that by now everyone's kind of sorted out how they're going to engage with Top Hat, whether that's through their web browser or through the app or through texting. Hope that you've resolved that, figured that out. If you haven't, let me know. I've uploaded and updated the scores for the first few days of Top Hat, and I wanted to, just get everyone to take the opportunity sometime today to look at the score in the Canvas gradebook and make sure that that is in sync with what they expected. If you if there's something where you're like, I was there on that day, I should have gotten points, just send me an email. I'd love to resolve that and reconcile that. Like I said at the beginning of last time, I did reverse course on engagement. I'm looking for participation. And so, in the event that you answer one or three questions, I'm looking to see you've done at least one question. So, I'm kind of putting intentionally a low bar, in part because I don't really want to. It's if you encounter a technical issue, um, I don't want to be the person who's like, or I don't want to say like, well, I'm sorry, not my problem, because I don't really believe that it is necessarily. I think that there are no fault circumstances, and so I'm not going to say like, not my problem. Um, I still have the questions that are going to be for homework. If you want to do those separately, that's your call. If you prefer to participate after the class has already taken place through the recording, by all means, go for it. Um, so basically, if you answer one of the questions, then great, that's what I'm looking for. Um, and so, at any rate, let's go ahead and proceed. So we're going to start the presentation, and we are talking about tertiary structure of proteins. Last time, what we were kind of closing the loop on was we were wrapping things up with respect to secondary structure, and we talked a little bit about predicting secondary and tertiary structure. Now, Alphabet, the parent company for Google, has a program known as AlphaFold, in which they are actively trying to basically um, increase the accuracy of predicting a protein structure from its primary structure. Now, that's really based on what we already know is possible with taking a primary structure and predicting a secondary structure. We're going to come back to that in just a second, but what I'd like you to know now is the tertiary structure of a protein. This is the folding of a polypeptide chain in three-dimensional space. So basically, how it wraps itself around anything in 3D is known as the tertiary structure. Now, the tertiary structure is concerned with the arrangement in space of all atoms within, this is a key part here, a singular polypeptide chain. Now, the conformation of these side chains, and that's a term that I want you to get familiar with. Um, Conformation, think about that as basically synonymous with shape. The shape of or the conformation of the side chains and prosthetic groups are included as well as the packing of the helices and sheets. Okay, so our helices and sheets, these are our most common our most common secondary structural elements. Now, this term prosthetic groups, um, is anyone familiar with a prosthetic group? At the molecular level. And I'll give you a little bit more of a lead before you answer that. Um, there is a prosthetic group that is very important for oxygen, basically distribution throughout your body. Um, so does anyone know what a prosthetic group a molecular prosthetic group is? Or can anyone think of an example of one? Now, an example protein that has a prosthetic group is hemoglobin. Now, when you hear the word hemoglobin or you hear the protein hemoglobin or myoglobin, does that ring a bell or does that kind of trigger you to think like, oh, maybe this is a prosthetic group? The prosthetic group that I'm kind of That's a great question, Milo. Iron is not a prosthetic group because of the affinity that the protein has to have for it. But when you think about hemoglobin and iron, do you possibly know the molecule that basically captures that iron within the protein? And I feel like whenever I say it, everyone's going to be like, well, I knew that if you had just asked the question in this particular way. The the group that I'm looking for is heme, Heme. Heme is an example of a prosthetic group. Heme is a example of a prosthetic group. And one thing that I want you to know about prosthetic groups moving forward is a prosthetic group of a protein or a molecular prosthetic group is just like a prosthesis that somebody gets as like a, an arm prosthetic or a leg prosthetic. It gives them an ability that they otherwise would not have. So heme gives proteins like hemoglobin and myoglobin a an ability to bind iron, which that's not really the unique ability, but instead the ability to bind oxygen is. And we're going to get more into that whenever we talk about protein functions. But when we talk about the tertiary structure, a prosthetic group is included as a part of that tertiary structure. Now, this is in addition to the packaging, basically the arrangement of any helices and beta sheets, alpha helices and beta sheets. Now, the formation of the tertiary structure is primarily determined by the interactions of the amino acid side chains with each other and the backbone atoms. Okay, so this is kind of building. If we think about our different levels of protein structure, we have our primary, secondary, tertiary structure. Without the primary structure, without peptide bonds, you don't have a tertiary structure. Without peptide bonds, you don't have a polypeptide, you don't have a protein. Okay. Now, without peptide bonds, you also don't have a secondary structure. So typically, whenever people talk about a tertiary structure, it's a convention that everyone's talking about R group interactions. So that would be, a arginine and a carbo and a aspartic acid, or an aspartic acid, a metal, and another aspartic acid. How those R groups interact with basically their surroundings as well as each other. So when we talk about tertiary structure, that's really what we're getting at is how these R groups interact with one another. Now, with that in mind, it's kind of it's relatively easy, fairly easy, it's relatively easy to predict the secondary structure of a protein, but it's much more difficult to predict the tertiary structure. Now, if you think about predicting the secondary structure, does anyone remember that plot that we talked about with the different angles that a peptide bond could take, or that could be the bond angles surrounding or adjacent to a peptide bond? Does anyone remember what that was? It starts with an R. It's a Ramachandran plot. Absolutely. Great job, Andrew. So Ramachandran plot gives you kind of a calculation of your acceptable or possible bond angles. And so when you have that, and then you have lots of data that we looked at propensity scores of different amino acids and where they're likely to be formed. Now all of a sudden we have, well, is this amino acid going to be in an alpha helix or a beta sheet? Or is it going to be in a random coil? When you have a certain number of, or when you have a large number of amino acids where you can say, oh, those are absolutely in an alpha helix, those are absolutely in a beta sheet, then you have a way to predict that secondary structure when you combine that with, well, let's look at a Ramachandran plot. Okay, but tertiary structure, man, that's a lot more difficult. Of course, you're using tertiary structure predictions based on tertiary structure predictions are based on secondary structures. Like you have, oh, well, there's probably an alpha helix here. So that means that this arginine is positioned here. But a tertiary structure of a protein is going to be considerably more abstract than a secondary structure. Now, with that in mind, let's go back to that secondary structure. This was our scoring system. And this is one of those I think that it's always important to remember different significant scientists. And we're going to talk about some here. Does anyone remember the authors of the review article that came up with this plot where they scored different amino acids? Chow and Fassman. Great job. So Chow and Fassman came up with this scoring system. And this was tremendous because then all of a sudden you could assign different scores, different amino acids. You could say, like, oh, okay, well, let's look at an alanine. An alanine, well, it has a high propensity to be found in an alpha helix. Okay, that's great. Now, what's its likelihood of being found in a beta sheet or a beta strand? That's a little bit lower. But then if we look at some amino acids, for instance, isoleucine. Isoleucine could actually be found in alpha helix or a beta sheet. It's got a decent propensity to be found in both. So this gives us a mechanism to make some sort of calculation and prediction. Now, this last one that I wanted to talk about right here, this last secondary structural element known as a reverse turn, you might also know this as, or might also see this referenced as a beta turn. Now, a beta turn is kind of interesting and unique because if we look at its propensities, glycine has a propensity of 1.77. Proline has a propensity of 1.32. Now, there are some other ones like asparagine is fairly high, but I highlight these two because if we look at the propensities, their propensities, absolutely, that's what, yeah, those are destabilizing. Those are our secondary structure breakers. So a beta turn is a unique place where those two amino acids are going to be found. That's not an alpha helix or a beta sheet. One thing that's really important to know is that proline and glycine, they have basically two different, or it's almost like they land at opposite ends of the spectrum of one another. Proline is too rigid of an amino acid to basically stabilize a secondary structure, whereas glycine is entirely too flexible to stabilize a secondary structure. No, but they will be found in beta turns or reverse turns. Oh, no, perhaps. Okay, yeah, there we go. Now, here's our first question. Which amino acid, so you guys can toggle back and forth on the PDF, which amino acid or the PowerPoint is most likely to be found both in an alpha helix and a beta sheet? Not one or the other, but which of the following has the highest likelihood of being found in both locations of the options that are presented here? 10 seconds. All right, let's check out the correct answer. Isoleucine is our correct answer. So I don't know why people picked proline or glycine. Those were the two amino acids that I highlighted as both being least likely to be found in secondary structures, specifically an alpha helix and a beta sheet. I'm not sure if maybe you pressed the wrong button or something like that, but proline and glycine were the two least attractive answer options. Histidine, it's a decent possibility, decent propensity, but proline and glycine, two thumbs down on those. Isoleucine is our most likely amino acid. Now, with that in mind, whenever it comes to kind of projecting a structure or saying, okay, well, is a secondary structure found? What you analyze is the primary structure. What I mean by that is you basically look at a polypeptide, A, I, I, L, I, L, A, and then let's go with an arginine here. So you'd look at a polypeptide, and every single one of these amino acids is going to get a score. And that score is that propensity score that we saw on the previous slide. And actually, I'll go ahead and throw a proline here, proline here, proline here. Now, when you look at this sequence right here from A to R, and you see those prolines, your first response should probably be, okay, there's a bunch of prolines there. Chances that any sort of secondary structure forms here has been tanked by those prolines. Now, if we eliminate those prolines, it's a little bit of a different situation. Okay, so we've got AILILAR. Okay, what do we know about these different amino acids? Isoleucine, it's got a high propensity for both a alpha helix or a beta sheet. Now, what about leucine? What about alanine? You're going to go through and basically cook up a score for each one of these. And then basically the kind of average or aggregate score that is evaluated is, well, is this higher for an alpha helix or a beta sheet? And if it's higher for a beta sheet, well, then a beta sheet's going to form. If there's basically a tie, if the overall propensity is equal for an alpha helix or a beta sheet, then it's going to trend towards an alpha helix. Now, the important thing about all of this is these are simply predictions. Now, with that in mind, though, let's think back to when we talked about the structure of an alpha helix. How many amino acids constitute an average alpha helix? Or how many amino acids are in an average alpha helix? Close. That's the number. So that's the number of amino acids. 3.6 is the amino acid. Billy, you're absolutely right. It's 10. So 10 amino acids constitute the average alpha helix. 3.6 is basically the number of amino acids that make one rotation. Now, I'm happy that you brought that up because one rotation does not mean an alpha helix, or sorry, let me rephrase that. One rotation, one complete rotation would be an alpha helix. Chances are, though, the average alpha helix is going to have about 2.5 to 3 rotations. So it's going to use about 10 amino acids. Now, with that in mind, if you're kind of scanning a polypeptide sequence, basically what you need is the better part of 6 amino acids. So we'd look at a window, you could phrase it. That'd be 3, 4, 5, 6. So we could look at these first 6 amino acids and we'd evaluate those. And then we would look at these 6 amino acids, evaluate those. Then these 6 amino acids, evaluate those. And you'd look at alpha helix likelihood, alpha helix likelihood, alpha helix likelihood, or beta sheet propensity. So you'd be evaluating these and you'd scan, 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 and scan. That gives you the possibility, or that gives you basically the opportunity to predict the likelihood of an alpha helix forming in any given location. Now, this is all what is done in the process of predicting secondary structures. That's why you might look at a secondary structure prediction tool, which it's easy enough. Just go to Google and type in secondary structure tool or secondary structure prediction, and you'll find any number of different tools. If you ever get the opportunity to take a bioinformatics class, if you're interested in protein structures, I would absolutely do that just because there's no shortage of protein data that's out there. And going from secondary structure to tertiary structure, that's the big thing. That's billions of dollars.
Oh, excuse me.
Yes.
Yes. Okay. Hi. How are you? I have a quick question here, really quick, if you don't mind. So, like, if you could go back really quick to like your the previous slide, if you wouldn't mind. And then you had that that whole diagram like drawn where you have like the the amino acids within the polypeptide.
Yeah,
I
so
had
I'll
a
just
question. Sorry.
ALILARL. Okay.
Uh-huh.
Yes.
Of course. Okay, so whether to determine kind of like the whole like the completeness of like the polypeptide, whether it's going to act as a helix or if it's going to show character within as a B sheet. So are we to for every specific per the amino acid, are we supposed to like declare it as helix character, and then for another uh a, amino acid were to declare, like what per like the like it's like nature, are we to declare it, as such, and then from there, or are we to take like account of how many like how many gosh darn it how many helixes and then how many beta sheets, and then from there we're to determine, oh yeah, this whole okay, gotcha.
Okay.
Okay,
So I
I
think,
saw you nod, so
well, no, I was nodding just kind of like, okay, I'm following. And I think that
okay.
I think I can answer your question.
Mhm.
Um, Andrew, are you asking if we have to predict if it's an alpha? I'm not sure who you're asking that question to. I'm not going to give you a polypeptide and ask you to predict whether its secondary structure or what its most likely secondary structure is. That would be something that you could do in a bioinformatics class. Um, the the closest that I would get to that sort of question would be like, okay, here's a polypeptide that's 6 amino acids long. Here's a proline in there, proline in there, proline in there. Which of these is most likely to form an alpha helix? Now, Adam, to answer your question, if I understand correctly, what you're going to
Uh-huh.
do is you're going to take this polypeptide, this A, and we're going to look at propensity for alpha helix or beta strand. And we're going to see, okay, well, this is 1.41. And what I'd do if we like graphically, the way that I'd do it is I would set it out as alpha helix right here. And
Hmm.
so I'll say propensity for alpha helix and then P sub beta for propensity for beta sheet. Alanine's going to get a 0.72. Then for leucine, I'm going to get a 1.22. And then for leucine for an alpha helix, it will be 1.34. Isoleucine for a beta strand would be 1.67. And then down here it would be 1.09. And so from a technical standpoint, and the part of this prediction is where you take every single amino acid and give them those scores. And so I would look at a frame of 6 amino acids. So here's my first three, four, five, six. And I would say, okay, the computer program that you write or the software, the script that you write for this, that one would write for this, would basically ask a couple of questions. The first question, is there a proline there? Answer is no. Okay, so chance that immediately pushes this to more likely than not to form some sort of organized alpha helix or a beta sheet. Then what we would do is look at every single amino acid and say alanine. Okay, in the first position, there's an alanine, and we have an alpha helix propensity of 1.41. And then the leucine is 1.34. Isoleucine is 1.09. Leucine is 1.34, 1.41. Arginine is 1.21. All of these scores are greater than one, then or for an alpha helix. So the chances that this 6 amino acids or these 6 amino acids form an alpha helix are pretty good. Then what we could do is compare them to the beta strand scores. Okay, so beta strand for leucine is 1.22, alpha or alanine is 0.72, arginine is 0.84. So we look at this frame of 6 amino acids and come up with basically a score, an average, and find that this, and I'm not a mental math master, but I would predict that this, let's see, we got three that are below. Well, this would be greater than one. And I would say it's like probably like 1.25 or so for the average amino acid propensity. And this would probably be like 1.05. And so if we're comparing these two, the likelihood of forming a beta sheet or an alpha helix, we would go with the likelihood is that this strand or this portion of our polypeptide is going to form an alpha helix. Now, does that answer your question, Adam?
No, that's exactly what I was asking for. Yeah.
Okay, great. And then this is a part of a process. So we looked at these six amino acids. And then what we would do, and I'm going to change colors so that I can verify this, is we would shift over, look at these six amino acids. And if, let's say that this leucine right here doesn't, or this next amino acid doesn't disrupt our secondary structure, that would continue to form an alpha helix. And then we would look at the next six amino acids. Does that form an alpha helix? The next six, and so on. And then, at some point, just by random uh, random amino acid positioning, you're going to find something that's potentially a secondary structure breaker or a cluster of secondary structure breakers. And a cluster just would mean like, 5 out of 6 amino acids are prolines or glycines or something like that. And so that's really what you're doing. And that's the value of this because you can have, and as we've seen, you can have a alpha helix that consists of like 5 or 6 amino acids, and you have those little coils. Um, does anyone else have any questions about that? Okay. So the next place that we're going to go to is another kind of valuable bioinformatic tool. And this is basically a scoring scheme that looks at the hydrophobicity of a given amino acid. So I think that this is, you know, propensity scores and hydrophobicity scores. I am not going to expect that you have these memorized. I expect you to understand what this is and the value of this. But what this table does, or what this scoring scheme does, was it basically evaluates every single amino acid R group and classifies them based on how polar or non-polar they are. At the bottom, you're going to see lysine and arginine, two positively charged amino acids, asparagine, aspartic acid, glutamine, glutamic acid, histidine. These are all polar amino acids. Now, at the top, we're going to see isoleucine, valine, leucine. I've always been a little bit surprised by proline because if I think about proline, I always classify it as a non-polar amino acid, but I think that it's kind of in this gray area because of the fact that the N termini of this amino acid is almost like still a part of the R group. So that's why proline has this kind of odd positioning right there. But what we're looking at is the higher the score here indicates the more hydrophobic that amino acid is. And now that's really significant because when you think about hydrophobic, water fearing, you think about something that basically wants to get away from water. And when we think about a protein, a protein folding into a shape that looks like this is our tertiary structure of a protein. Well, this right here, this side of the protein is exposed to the environment. So um, this is a exposed residue, or this is an exposed portion of the um, protein exposed to the environment, exposed to the solution that it's in. Whereas these amino acids that are kind of tucked deep in here, well, those are really packed in the center. And those amino acids are more than likely going to be your hydrophobic amino acids. This is also significant because we're always kind of curious about how different amino acids interact with their environment and interact with all aspects of their environment. We're thinking about this or we're talking about a biological molecule, a protein. How does that protein interact with like a membrane or something like that? When you think about a lipid bilayer, you've got polar head groups, non-polar tails. So arguably the internal portion of this lipid bilayer is pretty darn hydrophobic. So if you have a protein that spans a membrane, then that protein is going to probably have a lot of isoleucines, valines, and leucines right in here, in the portion of the protein that's in direct contact with that hydrophobic portion of the protein. So using a hydrophobicity plot or a hydropathy plot or a hydropathy plot gives you a way to look at, okay, well, is there a region of this protein that is densely hydrophobic? And if we look at this model on the right-hand side or this graph on the side, this is a score from -2 to 2.5. And on our x-axis, we've got our N-terminal down here to our C-termini of our protein. So this plot right here shows a protein that's approximately 15 or no, probably about 1100 base pairs. We've got a hydrophobic region right here. So between amino acids, let's call it 630 to 6, let's call that, no, let's call this 620 to 640. So 620 to 640. The relative hydrophobicity of the amino acids in that region of the protein is higher than elsewhere. Save this N-terminal portion of our protein. But what this is valuable for is it gives us a mechanism to predict, okay, well, we can compare this to secondary structure prediction. And then you can say, okay, well, this protein has a high propensity or probability of forming an alpha helix in this portion of in this region, and it's got lots of hydrophobic residues. When you use those combinations of data, then you can say, okay, well, what do I think this protein is? I think that this protein is potentially a membrane spanning protein. Now, if we were to look at something like this protein on the right-hand side and a hydropathy plot for that protein that's on the right-hand side, we would see something that looks a little bit like this. Here's our score, and then our amino acids. We would see something that hydrophobicity is kind of low, and then let's see, this would be 7. So we would expect 1, 2, 3, 4, 5, 6, 7. So we have 7 regions on our hydropathy plot that has a dense clustering of hydrophobic amino acids. That gives us a way to kind of predict and say, oh, well, based on the hydrophobicity of the amino acid distribution, then got pretty good reason to believe that maybe there's a couple of what are known as transmembrane domains. Then, if you were to look at a secondary structure prediction, and you found that there's a high probability of alpha helices here, 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 here. That's even more data or even more reasonable support for the idea that you have 7 alpha helices and you have 7 transmembrane domains. What this right here is basically all done before you actually look at that protein in 3 dimensions, before you do anything, before you do any sort of like wet bench work where you try to purify that protein. So that's the rationale for why you do these things, because the more information you get about the secondary structure, the more insight that you're able to put together or that you get for your tertiary structure of your protein. And there we go. Now, the type of protein influences a hydropathy plot. I think I would flip that around. I would flip that around and say the hydropathy plot as a piece of data it doesn't influence, instead it suggests something. Like you could draw a conclusion about the function or the type of protein. Darlene, what's your question? Do you have a question about this?
Yeah. I guess I'm not understanding what would make it like an alpha helix.
Okay. So so in order to kind of look at a protein, and please stop me if I, yeah, if you need to. Um, the every amino acid, our group, has different properties. Every amino acid our group can participate in different types of interactions. Some are going to be hydrogen bonders, ionic interactions. And what Chow and Fassman did, these two researchers back in the 70s, was they almost did like a survey. They said, okay, we're going to look at every paper where a protein structure has been resolved, and we're going to dissect it. We're going to dissect those proteins, and we're going to say, all right, well, within an alpha helix, what do we see? What amino acids make up that helix? And they came up with numbers. They came up with a scoring system. And so this is almost a matter of let's it's let's see where things are and assign a probability. And so we can use that. It's almost like there's a background information. It's almost like there's precedent. So what they found was that alanine and arginine, glutamine and glutamic acid, histidine, isoleucine, leucine, all these, basically any amino acid that now has a score of greater than one was an amino acid that they found pretty commonly in an alpha helix. And so that was the basis for this. So they looked at secondary structures. They looked at structures of proteins and said, all right, what amino acids are in an alpha helix? Oh, well, there's lots of alanines. We see alanine a lot. We rarely see glycine. Okay, so we're gonna, they came back and they classified glycine and proline as secondary structure breakers, so amino acids that are not likely to be found in an alpha helix. Does that make sense?
Yes.
Okay. So in terms of the likelihood of something being found, that was what Chow and Fastman were looking to do. And so what that allows us to do is then look at the primary structure and make a prediction. Oh, well, I think that there's an alpha helix here. So that was one part of what we wanted to do, or one part of like structural biology as a field. The next part was, all right, well, let's resolve this study a little bit more. And what we came up with was this hydropathy plot, where again, we're looking at the chemical characteristics of every amino acid. And then you see, okay, well, there are some that are more hydrophobic than others. And so we gave them a high score. And so my point of talking about these two things was it gives you a means to predict based on primary structure alone where a protein's going to be found. You can see, oh, well, there's lots of hydrophobic residues. What's a hydrophobic portion of a cell? Membranes. Membranes are super hydrophobic, or they have a hydrophobic region. So if you've got a protein that has a hydrophobic region, you got a couple of ideas. One, those residues might be kind of on the interior of my folded protein. Alternatively, they might be part of a transmembrane domain, like here, 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 or here. These are all like spanning that membrane. Okay. Now, that's that's all kind of like we've got more information. This is great. the more information that we have, we can put together a model or an idea of a tertiary structure. So the hydropathy plot, it's very okay. So I would say it's to a degree. So Morgan, I'm sorry I can't say absolute yes or no, but the more information that you have, the more you can use to kind of predict what a protein's going to do. So if you combine a hydropathy plot with the Chow and Fastman secondary structure prediction, and you see a pattern, then you can say, oh, well, there's a pattern where there's lots of hydrophobic residues that also happen to be alpha helices. I'm taking these two different data sets, the Chow and Fastman prediction and the hydropathy plot. Whenever I bring them together, oh, well, you know what? If I had to guess, these alpha helices, which happen to have lots of hydrophobic residues, I'm probably going to throw those as a part of a transmembrane domain. And if something's associated with a membrane, well, it could be involved in cell signaling. It could be involved in import of ions. It could be involved in a number of different processes. Does that help? Okay, great. So, so with respect to our kind of our beta turn, um, the third structure of or the third secondary structure is where you're going to find these prolines or glycines. And it's in this N position where you're going to find that proline or that glycine, um, where basically you have a very rigid or very flexible amino acid that enables that kind of abrupt turn of a polypeptide sequence. So proline or glycine are going to be found in those reverse turns or beta turns. So they're secondary structure breakers, but they also kind of shoehorned off this nice part of a secondary structure that they're participating in. Now, when it comes to a tertiary structure of a protein, this is a pretty good model of what a protein can look like. We've got in purple are alpha helices, in yellow are beta strands that make up a beta sheet, and then in blue we've got these random coils. Now, one of the things that I like about this structure here is look at this right here. There's one small subsection of this that basically makes up an almost alpha helix. So this cluster, this would probably be like two amino acids that have a high propensity for an alpha helix. Now, is this a complete rotation? No, it's not. But it's this cluster that you can classify because it's not just, or I would say 2 to 3 amino acids, where a portion of an alpha helix can form. And then all the blue region is random coils. Now, it's these random coils where predicting tertiary structure of a protein gets really hairy and really challenging because there's no kind of organization, or there's little organization to those random coils. I mean, that's why people decided, oh, we should probably call them random coils because they're randomly coiling. Our alpha helices, beta strands that make up a beta sheet, and then turns and random coils. There we go. Now, when we think about proteins, and specifically when we think about a globular protein as opposed to a fibrous protein, they're basically in two different states, an unfolded state and a folded state. Now, the folded state is your functional state where it can actually do something, whereas an unfolded state is pretty much non-functional. It's your polypeptide, but there's no kind of organization to this. Now, when you look at this, most proteins in living cells are going to be in that folded shape. And that's just because they found a low energy position that is that folded state. Now, proteins can be unfolded, and typically they're unfolded with a treatment. For example, heat. If you heat up a protein, what are you going to do? You're going to disrupt hydrogen bonds. You're going to disrupt ionic interactions, and you're going to denature that protein. This is the term denature is one that I want you to be familiar with. So heat denaturing. Now, we're going to skip this question. No, we'll do this question. When we think about different R groups or different amino acid R groups, which bond is not typically found between two amino acid R groups? I'm not talking about the backbone. I'm talking about those R groups. If you talk about like an arginine R group and a glutamic acid R group, what types of interactions are found there? And then which one is not typically found between two amino acid R groups?
I might be able to assign her. You want me to try? Yeah. All right. Bunch of numbers.
2 seconds. Right, our correct answer. Great job. This is one that I'm talking about the R groups, not the backbone. Um, Hydrogen bonds are pretty common. Ionic bonds are good examples of bonds that are found between different amino acids. Um, I would, if this is a question that you did not get correct, I would definitely look back at how I classify my amino acids. Peptide bonds are rarely, if ever, found between. It's possible, but it's just not very common. Um, so there we go. Now, with that said, what are some of the forces that stabilize our tertiary structure? Hydrogen bonds are pretty common between amino acids like serine and threonine, serine and histidine. Almost all of your polar uncharged and many of your polar charged amino acids participate in hydrogen bonding. And here's just an example. This would be an example. Well, this is just an example of hydrogen bonds, a hydrogen bond donor and acceptor. Disulfide bonds are found between two different cysteine residues. A disulfide bond is a bond between two sulfur residues. Now, this is significant, and I bring this up because it's so important. This is one of the major covalent bonds that exists between different amino acids or different amino acid R groups. It's not the only one, but there are post-translational modifications, so amino acids can be modified and changed after a protein has been translated. So these covalent bonds, this disulfide bond is an example of a covalent bond. Now, those disulfide bonds are basically going to be, well, they're going to be pretty agnostic to pH changes. They're a covalent bond. They can only be broken by extreme heat and extreme um, pH conditions, or they can have a reducing agent. So now here are your major forces that stabilize the tertiary structure. Hydrophobic interactions, ionic bonds, metallic hydrogen bonds. These are all great examples of forces that stabilize interactions. Okay, which of these R group interactions is pH change agnostic? So which one of these bonds doesn't care if the pH changes? If the pH goes from 1 to 1,000, it's not going to go to 1,000. Or another way of putting it is sensitivity. Which one of these is not sensitive to changes in pH? Now, there's one that's not listed on here, or another one that's not listed on here. Oopsies. Say interactions. Don't. And I know I kind of personified it there by saying caring, but that's the case. Oh, rats, I erased that too abruptly. Hydrophobic interactions are also, they don't care about pH changes. So let's look at our answer. I'm hoping that everyone got this. Disulfide bonds. Yep, disulfide bonds, they're a covalent interaction. Hydrogen bonds and ionic interactions can both be disrupted by pH changes. Okay, well, we are going to stop right here. We've got a couple more slides to pick up with respect to unfolding of proteins. We'll tackle that at the beginning of class on Wednesday. And then if you have any questions, please contact me. I'd love to talk to you about anything that we're talking about in class and clarify anything that you have questions about. I'm really happy with the conversation and questions that you guys gave today.